Hey class, let's get started with our discussion on microbial genetics. This chapter is a doozy, and in my perfect world, it'd really be two separate chapters. A chapter on microbial genetics and a chapter on genetic engineering. But your text decided to squeeze it all together, so that's what we're going to do as well. So let's start by defining some broad terms. What do I mean by genetics? Genetics, formally or officially, is the study of inheritance or heredity of living things. So when we look at our heritable traits, those are the traits that we inherit from the generation prior to us. So I, for example, have a widow's peak. I inherited that widow's peak from my parents. This study of inheritance explores a lot of different things, such as the transmission of these traits from parent to offspring, or how those traits are expressed. For example, my father has a blood clotting mutation. How that mutation is expressed in my siblings and I differs from individual to individual. Or we could look at the structure of the genetic material and the function of that genetic material. So is that genetic material going to be tightly bound? Is it going to be loosely bound? Is it going to be in linear or circular chunks of DNA? Are they big pieces or little pieces of DNA? And what is the function of that DNA? Not all DNA is genes or codes for genes. Some DNA has a purpose other than actively coding for a gene. And then finally, the study of genetics encompasses how that genetic material changes, how genes change throughout time, and how we can copy and duplicate those genes and then change one gene and then not change the other gene. So it's a wide, broad, open field. Usually genetics is going to be a 3 or 4 credit undergraduate class that most biology majors need to take. Within that broad umbrella of genetics, there's the subfield of the genome. The genome is all the genes, all of the genetic material, all of the DNA, all of the RNA, the rRNA, the mRNA, the tRNA. If it's made of nucleotides, it's included in the genome. All of the genetic material in the organism, the vast majority of that genetic material is going to exist in the form of chromosomes. In eukaryotes, those chromosomes are going to be in the nucleus. In prokaryotes, those chromosomes, or usually chromosome singular, is going to be in the nucleoid region. Every once in a while, we'll have little chunks of DNA in the organism. Those little chunks of DNA typically are referred to as plasmids. In eukaryotic cells, some other organelles will have genetic information. Those include the mitochondria. Yes, mitochondria have their own genome, mtDNA, mitochondrial DNA, and the chloroplasts also have their own genome, the ctDNA, or chloroplastic DNA. We don't spend a lot of time focusing on eukaryotes in microbiology, though, because we're mostly interested in those prokaryotic bacteria. In all those cells that have their genomes, I should also talk about non-cellular functions, or organisms. And I use that term loosely when I refer to viruses. A virus is going to have their genome as either DNA or RNA, depending on how that information is stored in the viron particle. So what is a chromosome? A chromosome is just a big piece of DNA. I'll say it again. A chromosome is a really big piece of DNA. Now, that big piece of DNA is going to have multiple functions. It's going to have multiple sections of it that have multiple things done on those multiple sections. Usually I like to describe a chromosome as a book. And when you pick up a book, there are multiple, like a cookbook, for example. A chromosome is the cookbook, and then each gene in the chromosome is going to be a recipe in the cookbook that tells you how to make a specific protein or a specific dish. And you can think of the genome as your cupboard full of cookbooks with all of the different chromosomes or all the different recipes arranged in all of the different books. For our eukaryotes, as I mentioned before, those chromosomes are going to be located in the nucleus. The DNA in a eukaryote is going to be really tightly packaged around proteins. And those proteins the DNA is wrapped around is called a histone. For many eukaryotic organisms, that DNA is going to be diploid, so there'll be pairs of duplicate chromosomes. Every now and again there'll be haploid, and sometimes there'll be octoploid or tetraploid, so on and so forth. Usually it's going to be in multiples of 2 if it's more than a single. And then finally, in eukaryotic organisms, chromosomes are linear, so they have 2 discrete ends. Bacterial chromosomes and other prokaryotic chromosomes are going to be circular in appearance, and they're always haploid. Bacteria always have a singular haploid genome, so they'll only have one chromosome. And instead of eukaryotic histones, the DNA are going to, in a bacterial chromosome, is going to be condensed into a pocket that's by a protein that's similar to the histone, but not quite the same. There are some significant structural differences that we are not going to discuss in this class. So, to summarize the slide, a chromosome is a big piece of DNA, and it has multiple genes within it. The genes within a chromosome can be broken up into three categories. We have structural genes. These are the ones that most people think about when they think of genes. So when I talk about transcription and translation and all that jazz, I focus on structural genes. These are genes that code for proteins. These are the best understood. We also have genes that code for RNA machinery, for protein production. These are genes that code for ribosomal RNA or rRNA. Those are fairly well understood. Sometimes these genes also will code for other ribozymes. Ribozymes outside of the ribosome are fairly rare, but they do occur. And then finally we have regulatory genes. These are genes that control other genes. These regulatory genes are very poorly understood. These are kind of the new kids of the block in terms of gene regulate, or in terms of studying genomics. We also have two terms that we need to describe or define for us. They are genotype and phenotype. The genotype is the sum of all genes. So the genes that you get determine your genotype, and the expression of the genes. As the genes are turned on and off, the traits that are expressed are called the phenotype. Your genotype will determine your phenotype. And usually at this point, this is when you have a long discussion about dominant and recessive genes, and you make some Punnett squares, and you have red flowers breeding with white flowers, and you get pink flowers and blended and codominance and a whole host of stuff that we're not going to focus on. So genotype is all the genes, and then the phenotype is the trait that you see. So here's the next summary slide. Let's look at the three kinds of organisms we're going to focus on. We have our eukaryotic cells, we have our prokaryotic bacterium, and then we have our virion particles. In a eukaryotic cell, we have chromosomes that are linear, and they are packaged in a nucleus. They're also going to be in the chloroplast, the mitochondrion, and then we could also have some DNA present in the cytoplasm in the form of a plasmid. I should also emphasize that the chloroplast and the mitochondria do not have chromosomes. Only the nucleus has chromosomes. The chloroplast and mitochondria have segments of DNA. In a bacterium, we have the chromosome that's circular in nature and it's located in the nucleoid region, and we'll also have plasmids. And then finally, in a virus, we'll have either DNA or RNA for their genomes. And as we look at the genome of an organism, it's very important that we are comfortable with and understand the molecules used to make up the genome. Those molecules, those monomers, are called nucleotides. All nucleotides have three basic subunits. They have a phosphate group, they have a deoxyribose sugar, and a nitrogenous base. And this is for a DNA monomer. If it was an RNA monomer, instead of a deoxyribose sugar, it would just be a ribose sugar. These nucleotides are going to covalently bond to each other. Phosphates bind to sugars, bind to phosphates, bind to sugars. So we'll make a sugar-phosphate backbone. Those sugar-phosphate backbones are going to be on each individual strand of DNA. So if we look at a coiled alpha helix of DNA, there will be two sugar-phosphate backbones. In between our sugar-phosphate backbones, we have those nitrogenous bases. And because there's a set sequence, a sugar to a phosphate, a sugar to a phosphate, we can have some directionality to the DNA molecule. One end of that sugar is going to be the 5' carbon end, and the other end is going to be the 3' carbon end. So when we talk about the orientation of DNA, we typically talk about its, or in terms of the numbering of the carbons of that deoxyribose or ribose sugar. So we'll have the 5 to 3' orientation. On the number 1 carbon on that deoxyribose sugar will have a nitrogenous base attached. That nitrogenous base attaches to the 1' carbon of the ribose or deoxyribose sugar, again, DNA versus RNA, with a covalent bond. The two categories of nitrogenous bases are the purines and pyrimidines, which can be further subdivided into our adenine, thymine, guanine, and cytosine. those purines and pyrimidines are going to be capable of base pairing with each other with weak hydrogen bonds. And I say hydrogen bonds as are weak because we're comparing them to the covalent bonds that hold the single strands of the DNA molecule together. Hydrogen bonds are relatively strong if you compare them to other kinds of chemical bonds like London van der Waals or van der Waals bonding or London dispersion forces. Those make hydrogen bonds look strong, but the covalent bonds and the rest of the DNA molecule make the hydrogen bonds look weak. Because we have these comparatively or relatively weak hydrogen bonds in that DNA molecule, we can easily unzip the DNA molecule and gain access to the information encoded by those nitrogenous bases. These are some base pairing rules that you must memorize. Most people have these memorized after their high school biology class, but it's important to review. Adenine will always base pair with thymine. A always goes with T. Guanine always pairs with cytosine. G always goes with C. So here we can see a generic structure of DNA. We have a phosphate, and then a deoxyribose sugar, and then a phosphate, and we have a 5 to 3 prime carbon. So that's our directionality. DNA is always read from the 5 to 3 prime direction. And then on the 1 prime carbon of our deoxyribose sugar, we're going to have a nitrogenous base attached. Here, since we have a G, I know that that is a guanine. C's and G's are going to have 3 hydrogen bonds holding them together. A's and T's will have 2 hydrogen bonds holding them together. That'll become significant later when we talk about PCR and melting temperatures for our DNA. So note that the DNA backbone is phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, in a 5 to 3 prime direction. So there's some directionality, and then in between our phosphate sugar backbones, we have nitrogenous bases linking up with each other via hydrogen bonds. This causes our DNA to have a twist to it and allows for our DNA to be easily unzipped and rezipped so we can access the genetic information and then store the genetic information. If we look at that alpha helix, that double-stranded chunk of DNA, it has an anti-parallel arrangement. That's a really fancy way of saying that the 2 strands run opposite to each other. I'm going to go back a slide here. We can see that we have one arrow pointing down from the 5 prime to 3 prime direction. And then if we look over here, we have 5 to 3 ascending. On the left side, it's 5 to 3 descending. On the right side, it's 5 to 3 ascending. We always read the genetic information in the 5 to 3 prime direction. So we have one direction descending, and the other strand is ascending. It's anti-parallel. The strands run in the opposite direction of each other. The order of the bonds between the carbon on the deoxyribose and the phosphate is used to keep track of those 2 sides, those 5 primed carbons to 3 prime carbons. And that's some organic chemistry nomenclature, where we have the 1 prime carbon, 2 prime carbon, 3 prime, 4 prime, and then 5 prime carbons on those deoxyribose sugars. This anti-parallel arrangement on the DNA is a very significant factor in terms of reading the genetic information and then synthesizing new strands of DNA. Because when we're making the new strands of DNA, we need to conserve this anti-parallel arrangement or take this into account. This process of making a new piece of DNA or going through DNA replication is called a semi-conservative replication. We call it semi-conservative because if we look at the new double-stranded piece of DNA, half of it is brand new and the other half has been recycled. This steps or these processes of DNA replication takes many different enzymes working together. If you take human physiology with me, we'll spend a lot of time talking about that in class and going over some of the enzymes involved. These enzymes will function to separate the DNA molecules. One copies and then the other will finalize the base pairing of those sister molecules. Each daughter molecule of DNA, I should say, is completely identical to the parent molecule in terms of the genetic information contained. So in terms of its chemical composition. These new daughter molecules are not completely new though. They're semi-conservative replication. One of the strands is going to be, that came from the parent, served as the template strand and has been completely recycled, and then the other strand is brand spanking new. So if I were to replicate this piece of DNA, one strand would be reused entirely in the daughter molecule. And this one strand that's reused entirely contains all of the genetic information, the A's, T's, C's, and G's, that I can base pair new nucleotides with to make my daughter molecule's new strand of DNA. Here are some of the enzymes involved with DNA replication. I'm not going to be super nitty-gritty or picky about which enzymes, other than I want you to know that DNA helicase unzips the DNA helix. DNA helicase unzips the DNA helix. That's one that's notable. You should focus on that one for DNA replication. Here's an excellent figure showing this semi-conservative process. We have in blue the original strand, and then down here we have the daughter strands. And you can see that half of the daughter strand is from the original molecule, and then we have in red the new strand of DNA for that daughter molecule. And because of the directionality of the DNA, one daughter molecule can be synthesized as one continuous run of DNA. And because of the directionality, the other daughter molecule can only be synthesized in little segments of DNA. These are called Akasaki fragments. We aren't going to spend a lot of time focusing on those between the leading and lagging strands. This is not a cell bio or molecular bio class. Just know, focus on that when DNA is replicated, one strand can be synthesized as one complete molecule, a leading strand, and the other strand can be synthesized in little segments. This is the lagging strand that's going to be synthesized in segments. The leading strand can be synthesized as one continuous DNA molecule. As we are synthesizing our new DNA molecules, those new DNA molecules are going to have a start and stop sequence. As the bacterial species you're working with are going to be synthesizing their DNA molecules, they can do this at an incredibly fast pace. They can do it at about 750 base pairs per second at each replication fork. So that'd be the same as typing 750 characters a second. I don't know about you, but I can't type that fast. If we look at DNA polymerase I is going to remove an RNA primer and replace it with DNA. The ligases will move along the lagging strand and link the fragments to complete the synthesis of the two daughter molecules. So if we go back a second page here, we have these individual daughter segments here on the lagging strand. DNA ligase will link them together. During this DNA replication, mistakes do occur. If you are synthesizing DNA at 750 base pairs per second at each individual replication fork, there's going to be a mistake every now and then. But we have some wonderful proofreading mechanisms. And those proofreading mechanisms help to minimize mistakes. Mistakes are typically going to occur once every 10 to the 8th power or 10 to the 9th power bases. So about once every 100 billion to 1 trillion nucleotides, there'll be a mistake. Most of those mistakes, though, that happen between every 100 billion and 1 trillion nucleotides will be corrected by DNA polymerase III. It has some proofreading mechanisms that are built in. Here we can see for a bacterial chromosome, as that's being synthesized, it'll have two replication forks. We call that a replication fork because we look at the parent strand and then the two daughter molecules. It looks like a two-pronged fork to some people. To me, it looks more like a lambda symbol. As a bacterial chromosome is being synthesized, there'll be two replication forks moving in opposite directions, and then eventually there'll be a small segment where the two bacterial chromosomes are linked together. That's called the nick. At that nick point, the two daughter chromosomes can be separated from each other. Once these daughter molecules have been made for bacterial chromosome replication, we can now start the process of mitosis and then cytokinesis to divide the cell. So, class, concept checked. When DNA is replicated, we have two brand new nucleotide strands produced. If you don't know the answer, that's okay. Pause the video, check your PowerPoints, check the textbook, or rewind the video. 1, 2, 3, 4, 5. The answer is false. When DNA is replicated, we do not have two brand new nucleotides. We have a semi-conservative replication process. So we'll have one brand new and one recycled nucleotide strand in that DNA alpha helix. Boom, boom, Here we go. So when DNA is replicated, one of the strands will be old and one of the strands will be brand new. That's all we have for this presentation. If you have any questions about it, please post them to the discussion board or shoot me an email. Happy studies!